Hello everyone. In this video we'll be covering the basics of DNA replication. The main points that I will be covering are shown here on the left. Semiconservative model, basics of DNA replication, how it happens and the main proteins involved, how the leading and lagging strand works, and then things that are specific in DNA replication to prokaryotes and eukaryotes. Let's get started. First is a semi-conservative model of DNA replication. In simple terms, this means that the original strand of DNA is used as part of each of the new strands. Think of this analogy. Jimmy and Jake are two kids who love to build Legos but are currently fighting over who gets to keep the Lego house that they just made. Think of the Lego house as the original strand of DNA. One half of the house is one single strand, and the other half of the house is the other single strand, and they're hydrogen bonded in the middle. In order to break up the fight, their mom splits the house down the middle. She gives one half of the house to each kid, and then each kid builds the other half of the house. So here we have one half the original DNA, here we have the other half the original DNA, then each kid built the new half to the house, or the new DNA that's built. This is the same as how DNA replication works. Don't forget that DNA is a double helix. The two strands of the deoxyribonucleic acid are complementary to one another. When the cell's dividing and it needs to replicate its DNA, it splits right down the middle. You can think in this analogy that the house is one strand of DNA and each half is one single strand of DNA, as you can see here. Next comes the basics of replication. In order for replication to start, the DNA must be unwound. This unwinding starts at the origin of replication, which is a specified place on the DNA. To start, a protein splits the DNA apart and makes a gap called the replication bubble, as seen right here. Here we can see this circle DNA, where each gray line represents one single strand of DNA, meaning the two strands together makes a single strand of DNA. As in, if you were to split this down the middle, you'd have two single strands of deoxyribonucleic acid. The hydrogen bonds are in the middle right here, where you see the white line. Each place that the DNA is splitting apart right here are called replication forks. Next is DNA polymerase, which is arguably the most important enzyme. It builds the new strand of DNA on the template strand. The DNA polymerase contains many components. One of these is a sliding clamp, a part of the protein which keeps it bound to the template strand of DNA. Another component is the helicase, as seen here. This is the part of the protein that unwinds the DNA strands. In order for the DNA polymerase to work, there must be an RNA primer already on the template strand. This RNA that is put on the template strand first is done by an enzyme called DNA primase, or more specifically, DNA polymerase 1, and that is seen right here, primase. The DNA polymerase that does the building of the DNA itself is specified as DNA polymerase 3. One other protein used in DNA replication is topoisomerases. These enzymes are used to keep the DNA from winding up on itself as the helicase splits apart. It might be hard to picture, but as the DNA is being unwound, the tension over here builds up and causes this part to become actually wound tighter. And topoisomerases release that tension, make sure it doesn't get too wound up. One very important aspect of DNA to consider is that it is anti-parallel. One strand is arranged 3' to 5', while the other strand is arranged 3' to 5', and the other strand is arranged 5' to 3'. What I'm about to say is very important. When building DNA, DNA polymerase can only build DNA in the 5' to 3' direction. Look at this unwound DNA here. The top strand of the original DNA, commonly referred to as the template strand, is arranged 5' to 3', 5' to 3'. This way, the DNA polymerase can build the strand 5' to 3' underneath it in a continuous manner. They're always going to be opposite. So if you have a 3' here, you're going to have a 5' underneath it building that way. You have the 5' here, you have the 3' here. Always think match 5 to 3. The strand that can be built continuously in this way is called the leading strand. The bottom of the template strand is arranged 3' to 5'. You can't build the strand the same as up here, 5' to 3', because you'd have the 5 and 5 matching up and the 3 and 3 matching up as if this line were down here. That doesn't work. So, the DNA polymerase has to build it in segments, where it goes 5' to 3', 5' to 3', 5' to 3', because you can't go in a continuous manner up this way. These small segments that are being created are called Okazaki fragments. The gaps you see here in between the pieces of DNA are glued together by a protein called DNA ligase. Prokaryotes replicate DNA in a very simple manner. Their DNA is held in the cytoplasm of the cell and it's replicated right there in the cytoplasm. They have this simple circular DNA which has one origin of replication. This makes studying their replication process easy. Eukaryotes are different. They have linear DNA stored in the nucleus of the cell. They have so much more DNA than prokaryotes that they actually have hundreds of thousands of these origins of replication. DNA polymerase has the ability to start replication at all of these places nearly simultaneously. If there was one origin of replication, like for prokaryotes, for all that DNA, it would take way too long for replication to occur, and cell division would be nearly impossible. So, to recap what was covered in this video, DNA replication is semiconservative. Part of the original strand is used in each of the new strands. DNA replication begins at the origin of replication and is carried out primarily by DNA polymerase III. Don't forget that DNA polymerase III has numerous parts with specific functions in order for this to be carried out, which were covered in this video, specifically the sliding clamp, helicase, etc. DNA can only be replicated in a 5' to 3' direction. As a result of this, there is a leading and lagging strand of DNA. Prokaryotes replicate DNA in a simple manner because there is less to copy, while eukaryotes have much more DNA and replicate it in a more complex manner in the nucleus. Have a great day.